So good evening. Good evening. For those of you who don't know me, I'm Tim O'Shea. I'm the Principal and Vice Chancellor of the university. It's an enormous pleasure to welcome you to Professor Douglas Armstrong's inaugural lecture. Particularly delighted to welcome his wife, Pamela, his mother, Vi, his aunt, Vi, his father, Walter, his sister, Alex, his father-in-law, Harry, and his uncle, Alex. So that's a bit of bunching there. Douglas started his academic career in Glasgow, and I'm always very interested to know, what prompted people and how they got into doing what they were doing. And he was just telling me that when he was an undergraduate, he worked on flies because you could just do things very quickly, unlike mice, which take time to breed and such. And when he was an undergraduate, he established the pattern that he has today, which is gene sequencing in the daytime and computer programming to do the analysis at nighttime. And that's a very and that is a very consistent thread through his work. So having got this enthusiasm as an undergraduate, he did his doctorate in genetics at Glasgow, postdoc, and was firmly then set as a multidisciplinary researcher and somebody who's also working on getting patents and working with spin-out companies. He spent some time at Rice University in Houston, Texas, a very nice part of the world. I spent some of my career in Austin, Texas. And then in Edinburgh, he came here in late 2001. Massively successful academic, big framework program, 7 grants, big MRC grants, very important publications in journals like Bioinformatics, Cell, Nature, Molecular Systems Biology. As you would expect from a very successful academic, he's got a thriving group of postdocs and PhD students, but also exemplary record in industry. And I used to be the chair of stakeholders for the Scottish Enterprise Proof of Concept Scheme, and Douglas won 2 of those, which are highly competitive and very good ways of starting on. Lots of very good work in outreach, and we now have him both as a personal chair in systems neurobiology, but also part-time chief scientific officer for the company Brainwave Discovery, and part-time chief scientific officer for Actual Analytics. So please join me in welcoming Douglas to give his inaugural lecture.
Thank you very much for the introduction. I should point out that throughout the talk this evening, I'm going to highlight various different people's works, friends, collaborators, people who've worked in the group, and so on, and thank them individually. But I very rarely get the chance to say a broader thank you for all the support that I've had from both within informatics, from Centre for Integrated Physiology and the staff and students there, as well as at Rice University and at Glasgow. So I know several of you are here today from far afield, so thank you very much for coming. But also wanted to say thank you very much to the friends and family who have supported me over the years and put up with my work getting in the way of holidays and various other things. So when I've had to scoot off and write a grant or write a paper or go to a meeting in the middle of holidays, thank you very much for putting up with that. Even to the extreme of putting up with phone calls en route to the maternity ward at Glasgow University, Glasgow one year. The person who made the phone call, I think, is in the audience, but I won't single you out just yet. I think you know who you are. So, I would like to start off with a little bit of an introduction into what systems neurobiology is, because it's usually the first thing people ask me, is you're a professor of what? So I want to start off with a little bit of an overview of at least what I think it is and why I think it's important and why are we doing this at all? Why are we here? It's really about, or the driving force behind it is really about how brains work, and more importantly, how do they work normally, and what happens when they stop working, and how does that impact on people. And I don't want to be too much of a depressive about this, but it's a massive problem. 1 in 3 people are affected with some form of neurological disorder through their lifetime. It's the largest single cost to the NHS. When you add the social care and society costs on top of that, these are massive numbers, 77 billion in the UK. You're getting towards the numbers that politicians talk about in national deficits. So these are huge numbers, huge percentages of what we spend. And that's just looking at the financial cost. That's not looking at the individual cost, the burden on individuals, their friends, their families, and so on. And it's particularly bad in neuroscience. The latest World Health Organization's numbers try to estimate the impact it has on people's lives. 40% of years affected by disease or shortened by disease are some form of neurological disorder. It's the single biggest health problem that we actually have to deal with. There are lots of other important ones as well, but this is actually really the biggest one. So why don't we do something about it? The problem is it's incredibly complicated. It takes about 13 years to take any new development in research. So when you see in the newspapers, oh, we found a new gene for schizophrenia or a new breakthrough, it takes 13 years to go from that breakthrough to a drug that is actually being prescribed by a GP, on average, sometimes longer, sometimes shorter. It costs $2 billion to get there, so it's not cheap. And the failure rate, the attrition rate of those drugs getting to that stage where they can actually be sold and actually used to treat disease is incredibly high. It's much higher. It's as high as the worst of any of the other diseases. So there's a massive need, massive demand for better understanding the nervous system to lead to better drugs to treat diseases. So that's really where we're, the driving force behind this, behind what I do. But it's embedded in, as I said, a system which is incredibly complicated. So if you look at the brain, we have the structures in the brain, the nerve cells, which are the key parts, send a signal from one cell. So this is a cell coming through here. This is the axon of the neuron. There's an electrical signal comes along here, gets converted into a chemical signal, it jumps across a little gap, and then gets interpreted by a molecular apparatus here and converted into a new electrical signal that goes along. That's really how the nervous system works. There's lots of complexities in here. These things modify themselves, they change their connections, they learn, they change with age, with experience and genetics and so on. But fundamentally, this is what it's about. This little process here of these chemicals being released from one neuron, jumping across and being interpreted at the other. So sounds reasonably simple, but it's embedded in a much more complex structure. So you've got this molecular level at one site, so these molecules moving around. They're embedded in these synapses, which is this whole structure together. Those in turn are making networks of these neurons. So one neuron here, one neuron there. It's a connection between the two that are then getting more and more complicated and interconnected and going up to brain and ultimately behavior and even social interactions. So if you want to treat disease, you're looking at diseases manifest themselves ultimately in behavior. So it's how the individual changes their behavior. Whether it's a depressive illness or an illness that affects the movement or whatever, you're looking at behavior. But unfortunately, what you're really looking at is something that's changed down at this level here. So there's something going wrong in this signaling here, which either means this connection is broken, or it's modified, it's more active, less active, or it's inappropriate compared to usual. Now I'm going to talk in a bit of time about how to try and build models for these sorts of processes. So how do you try and simulate or build a model of your understanding of this whole series from molecular up to behavior? There's some extremely good work being done on how to do models at the network, super network and brain level. But in general, most of those networks don't go down to the molecular. So a lot of computational neuroscience is done at this level here. So you treat it as a network and you just basically say there's a little process, things jump from here to here. And it just happens. You approximate it into something simple. The problem is, if something goes wrong, it goes wrong at this molecular level, which has been missed by these simulations, by our understanding. And if you want to treat it, you use a drug from a pharmaceutical company, which typically will diffuse or get absorbed into the body and go into one of these things, bind onto one of these chemicals and make a slight change. So the disease starts here, its treatment starts here, yet when you look at brain models or simulations, which would typically be done in institutions like this one, they tend to ignore what is actually happening here. That's not a criticism of what they've done. Trying to bridge between these two is not easy. In fact, it's incredibly difficult. But we need to start developing the tools and the approaches where you can actually build this level of complexity into the bigger framework. That's what I'm going to try and tell you a little bit about this evening. Just to put it in context, this is an image done by an artist in New York that I found on the web that kindly lets me use it for talks. But actually a real neuron is a little bit more complex. So this is just a single nerve cell, and the little green dots on it are these synapses, these chemical systems that connect from this one neuron in orange here. Well, each one of these little green dots is a connection to another neuron. You can't see the other neurons because if they were labeled up, you wouldn't be able to see anything at all. This is a single nerve cell. Each one of these little green things is one of these little complexes here. It's one of these things here. So you're starting to get a feel for the complexity. That's one neuron out of the billions that are in a human brain that we've got to try and deal with. Okay? So 10 years ago, just as I started at Edinburgh University, we started a long-term collaboration with Seth Grant, who I believe is in here, looking at what actually makes these green dots up. So what is in there? Can we actually start to tease it apart and to find out how it's organized? His group developed expertise in actually isolating these from the brains of rodents and actually trying to characterize what is in there. And then we came along and helped with building the kind of databases and the informatics and the tools for actually assembling these into complexes. The process we go through is what we really call systems neurobiology. So we start off with what is relatively straightforward computing science, so data bioinformatics. So we assemble databases of information about what we already knew about the system that we're studying, how it interacts, and also then the new data and how you can potentially piece it together. We then build models, so what we think is our best understanding of the interactions between what is there. So this gives us a part list. This gives us our first rough map. So each one of these would be one of the molecules that we would find in the nervous system. Each one of these little lines is how it connects together. It gives us a kind of feel for what could connect to what. A kind of interaction map is what we would call it, or a protein interaction map. We then go into slightly more complex models, which you may be glad to know I'm not going to go into in too much detail tonight, which actually capture not just the interaction between each of these molecules, but the rules in terms of when they can interact, how they compete with each other, what happens if a drug binds onto one of these, what happens if a mutation in a human affects the affinity for this one with one of the other ones. So we can build that level of knowledge into these simulations. So when we started more or less 10 years ago, we built our first molecular networks of the nervous system, and they look like that, and they tell us obviously everything about the brain. Unfortunately not. So this was just the very first model we ever built. So this is just pulling together absolutely everything we could find in one of these molecular synapses. So I've shown you the little cartoon of it. I've shown you the neuron that has the thousands and thousands of synapses. And this is our best guess, or this was our best guess 10 years ago, of what was inside just one of those. So you've got thousands of these in billions of neurons, and each one of them is comprised of something which kind of looks like this. I mean, this is an approximation. It's not absolutely accurate, but that's the kind of thing that we're pulling together. And each one of these blue blobs there is a different protein, which could be a drug target, or it could be linked to a disease. So to pull back a little bit and look at a smaller subset, we focused in on a subset associated with a specific class of neuron receptor. Now, I don't want to go through this in too much detail. What I really want to do is just highlight, there's a list of gene names inside, or protein names down each of the columns, and there's a bunch of color codes, two of which you can ignore, the yellow and the green. We're not going to go through them today. But the key thing is the ones in magenta, the purpley color. Those are the ones which even 10 years ago had some sort of association with a psychiatric disorder. So not just neurodegeneration or anything, a psychiatric disorder specifically. So just a little subset of proteins that we found in a specific class of neurons, we got 30% of them had already been implicated in neural disease, telling us how important these subsets really are. So we really need to understand how these things are put together, how they connect, if we want to understand how to do the kind of next generation of drug discovery. Things have moved on a little bit. The models are getting bigger. They're getting more complicated. They're getting better connected. This is just one of the ones that we published last week with a bunch of collaborators in Amsterdam with Huss Smit and groups in Berlin. It was assembled by Oksana and Colin in the group. So these models are growing all the time, and they've now given us a kind of global picture of what is there. Those are just the map of potential interactions, what can happen. We've also started, as I said, going into these more complicated models where we've captured the rules as well. So in other words, these molecules can all exist, but they compete in the space, so they displace each other. And so we can capture that now. And we can also say that, for instance, these ones up here have to be at the surface of the structure. These ones in here can be below the surface and can move around with respect to each other. The big new thing that we've kind of done, and this is in collaboration with Vincent Danos' group, is look at ways where we can spatially constrain these and the interactions between them. So we can now say this molecule here is active only when it's connected to the ones in the same space as it. So we can actually start to build little 3D models of what we think is happening. So this is a 3D model of the little green dot you saw in the neurons. And it's very stylized, obviously. It's a kind of regular shape, whereas the real shape is obviously much more complex. But the key thing is that the position, or the relative position, of the coloured spheres, which represent different classes of molecules, is driven from real data. So we've worked with biologists to find out what the rules are for how these things can move around and what they can interact with. And then the simulation effectively puts these in there in the same way. We can then do this over time, so we can look at the availability of a molecule within a cell and how it propagates over the surface of it. And then at the surface here, this is obviously the part that would interface with the other neuron, look and see how the activity of this surface is changed with respect to the availability of different molecules. This is just a steady state model where the availability of everything is the same. But you could start to simulate, for instance, what would happen if we used a drug which stopped this purple sphere, the AMPA receptor, interacting with the scaffolds underneath. So we can actually now start to do the simulations which go from the gene level to the synapse level here, which actually tell you what the activity or the sensitivity of this is. So we can actually start to jump up that level. So that set of models and that set of approaches basically allows us to start to bridge from the molecular up to the synapse and up to the network level. So we can now start to simulate what is happening in here and how it might actually affect the behavior of this whole group. There's other groups working on similar approaches. It's not completely unique. But this is effectively what we would class systems neurobiology. It's this ability to go from starting at one level of biological organization and start to go up through the other ones. So that's systems neurobiology. How far can we take this? So as I said before, there's an awful lot of work in terms of the models at these other levels. I don't work in that area. I'm not going to go into them in any detail, but it does exist. It's there. There's a strong research community working in it. Can we go from there and actually build a model of, let's go for the big one. Can we build a model of a human brain from that? Well, okay, theoretically we can jump these levels, but we've got some fairly big numbers to deal with again. We're getting back into the sorts of numbers that bankers like. So 20 billion neurons, 100 trillion synapses. Yes, we might be able to simulate bits of this picture, but we don't know a fraction of it yet. That said, the Human Brain Project has recently just been awarded. It's a 10-year flagship project from the EU with the aim to do just exactly that, to try and scale up the simulations to this scale. How far they go in terms of simulating a real brain is a different question, but the key aim of this is actually to get the simulation scale up to that. Now, this is not just a challenge in terms of understanding the biology, in terms of working out the billions of neurons and connecting them up. It's actually a hardware or an electronics challenge as well. If you look at the number of connections and processes you have to simulate to get to that scale, you go way beyond anything that exists in computers that are available in industry just now. In fact, some of the calculations that were used in the early phases of putting the Human Brain Project application together took existing supercomputers and said, what would happen if you scaled them to full human brain simulation? And what would the power supply need to be like? The electricity cost is roughly the same as the gross domestic product of the UK to do it with current computational power. It's an approximation, but you can see that's just not going to happen. There's going to need to be huge breakthroughs in computation to make this possible. Okay, so Human Brain Project can come later. It's good to see there's some funding to try and do it, but it's not going to happen anytime immediately. So what about studying simpler organisms, and what can they tell us about more complex ones? So just to mention, obviously, the next one down potentially would be the mouse. That's used for a lot of pharmaceutical research, so it would be a good target to go after. So what would be involved in making a complete mouse simulation? Well, actually, it's a bit more tractable. You're into the realms of kind of high-end, very expensive supercomputers in terms of the scale of the problem. So with some of the larger IBM Blue Brain machines, you can actually get simulations of the right scale of a small mammal, and it's been done before. In terms of actually doing ones that are constrained by the biology, I think that's yet to come, and I think that will happen in some of the early phases of the Human Brain Project. Again, you do still have the power problem in that these things are expensive to run. So I'm not going to do that anytime soon. So I think what I really want to try and get onto is this beastie, which is what I've been working on for the last 20 years, Drosophila. So inside this is something a little bit more tractable. And this slide is a few years old now, so I'll update it in a few minutes' time. But basically, the scale of the computing problem of a fly brain, which you think should be pretty trivial, is at least tractable with modern computing. You could technically build one. I'm going to try and show you why that is possible, and also tell you why you might actually want to do it. You know, we're not just doing this because we want to do it, but because there's actually useful things we can learn from it. So that leads to my question for this evening, looking for answers inside the mind of a fly. So I want to cover from this point on two main issues, one of which is that building a fly brain is feasible. It's feasible now, and I want to show you what we've tried to do to take us towards that phase, and show where it's then going next. It's not finished yet. And then secondly, through that kind of understanding, the increased understanding we have of something like a fly brain, what can we then actually do with that that has got potential impact directly on human health? Because ultimately, that's why I started with. That's the important part. If it doesn't come back to that, then I'm probably wasting my time. So, just to introduce the fly. Some of you know it well. Some of you have seen it less. This is the house fly. This isn't the fruit fly Drosophila. This is the house fly. So you've all seen that before plenty of times. Drosophila is the little one down there. Okay, so we're looking at, take a house fly, shrink it down an awful lot, and we're talking about the brain of this little thing here. Should have brought some with me actually, but I forgot. It has an interesting life cycle. So there's a daddy fly and a mummy fly. And after they're about 24 hours old, you'll get the first viable eggs. And after about 24 hours again, the first larvae will crawl out. And they're essentially eating machines. They're fairly simple. Basically, their idea is to increase body mass until they're big enough to support the adult animal. They have a basic nervous system, so they eat until about 5-6 days old, until they're big enough. They go into a pupal phase for a few days, and then they hatch out into another pair of flies. The whole process from start to finish takes about 10 days. So with 2 flies on day 1, after 10 days, you've got the next generation, which could be 20 flies, 100 flies after 2 weeks, and then 100 times that after another 2 weeks. So if you want to answer biological questions quickly, fly is the place to do it, because you can still do this. Okay, that's what it looks like in terms of biology. What about its brain? It does have one. That's usually another common question. So I'm going to go through a few reasons why this would be interesting. So the first three are really from the biological perspective. So why is this interesting? Why would we do this? The second three are really from the more computational perspective. Again, why is this interesting? But for different reasons. First one's the nervous system, the brain of the fly. It's a movie from a collaborator of mine, Kei Ito. So this shows an animation of half a fly brain in 3D. It has 100,000 neurons, a little bit less than the billions that you'd find in the human brain. So it's a smaller, more tractable number. It's a number that most of us are used to handling. You're getting towards mortgage-style numbers. 50 synapses per neuron, so the little green dots on each of the cells. It has approximately 50. One of the interesting things about this is it's actually been measured in flies. So it's estimated for lots of other species. So people have put random numbers, not quite fully random, but they've estimated how many synapses there are per neuron. For flies, we know for large numbers of those neuron classes exactly how many neurons there are. It varies from individual to individual. We have got, through collaborations, and I'm going to come on to that in a minute, over 18,000 of these neurons individually traced in 3D and available in databases. In fact, we've got copies of them in hard drives upstairs. We also know what most of the sensory and motor neurons are. So we know how information gets into the brain, and we know how actions get back out. How does it control its muscles? How does it move around? We also know that it's not completely and utterly hardwired. So it does react to its environment. It does learn. It does change the way it's wired in a similar fashion to the way a mammal would in response to what it encounters in its environment. And we know the development of it. So we know where every class of neuron comes from, the developmental history of them. In fact, most of what's known about human development actually was initially discovered in fly development. How you've got the right number of vertebrae comes from the same genetic program or a similar genetic program to the different segments that are found down a fly back. Those are all conserved programs. So conserved molecular function, okay, fine, it's there, it's simple, it's tractable, and it has a nervous system. But what about, is it relevant to human at all? And I'm going to come back to this a couple of times through this evening. So to do this, one of the key studies that we were involved with, again, was part of this proteomics study that I discussed earlier, where we were mapping these protein complexes. So the protein complexes I showed you at the start in the kind of introduction to systems neurobiology were all from mouse brain. So, fine, mouse has a brain, fly has a brain. What's the differences between the two? And again, this is work that we did with a PhD student called Bilal Malik in collaboration with Seth Grant and Richard Eames, these groups. So in terms of evolutionary history, humans up here, and you can see chimpanzee, various, the primates up here going in through the mammals, mouse, rat, and the distance on this line shows the time in millions of years that you have to go back to the last common ancestor. So back at this branch point down here, there was a simple little mammal that gave rise to all of the others. To go to fly, you've got to go all the way right down here and all the way out there to get to fly, Drosophila melanogaster. So this branch point down here, where invertebrates, or the flies, worms, and so on, split off from the vertebrates, was about, it depends on which number you use, but 350 to 650 million years ago. So it wasn't exactly yesterday. So a long time has gone since there was some small worm type thing, some of which gave rise to all the insects that we have now, and some of which eventually gave rise to us. Okay? So 670 million years ago, there was a small worm, and it diverged in two. But these have nervous systems, these have nervous systems. The little worm presumably had nervous systems as well. So we want to go back and say, what was the similarity? So we can do that computationally, or we can estimate it computationally by just basically saying, what did we find in the mouse, which is in this column here. So mouse is 100%. If you say, can you find all the proteins we found in mouse? Yes, you can. You can find nearly all of them if you say, can we find it in human as well, just by looking at the databases of proteins, and yes, you can. And so most of the mammals have all the same numbers there. The interesting bit was if you looked at fly, in fact, not just fly, mosquito, bee, worms, they're all pretty much the same. you lose about 50% of the proteins that you find in the mammal nervous system. So it's simpler, it's missing a bunch of things. Interestingly, although it's 50% of the number, it has all the basic classes. So if you need 10 types of protein to build a synapse, mammals might have 20 of them, flies have got 10. But they still have the same ones. The same building blocks are there, there's just less diversity within that set of building blocks. We did the converse experiment because obviously one thing it could be is this missing number could just be the 670 million years of evolution that happened from the worm to the fly, which was different from the fly to from the worm to the human. And actually it's still the same, we still get the smaller number. So we've looked from both from the bioinformatics perspective and from the biochemistry. So it's there. It's a simpler nervous system, which is good. If we want to model it, we want it to be simpler. It's more tractable, but it has all the basic building blocks in place, and they're still recognizable in the fly as they are in the human. In terms of disease, there was a colleague of mine several years ago, Larry Reiter, built a database called Haemophila. This is nearly 10 years ago now, where he took every single human disease gene and said, can we find it in fly? So of 3,000 genes at the time that were linked to human disease, he found 2/3 of them in the fly genome. So there are things that are missing. There are diseases which don't translate into invertebrates, and that's not particularly surprising. It's probably more surprising that so many do. Now, we've revised these numbers. Jinan has a PhD student here, and Ian Simpson, used to work with the group now at BIOS, have revised these numbers, and the trend is the same. So there's no new breakthrough. The conservation in terms of disease genes is exceptionally high. So we've got a simple brain that's tractable. It's conserved molecular functions, so it's valid. It can tell us something about human nervous systems as well, and excellent genetic links. In terms of more computational ones, why would you be interested in fly? There's a couple of interesting reasons. First of all, there's a lot of interest from the computing and engineering areas in how you actually build autonomous learning organisms. So organisms which can decide the best route to solve a problem by themselves, that are adaptive to changes in the environment and that ultimately are low power. You don't get much lower power and more autonomous than flies figuring out what they're going to do. I'm going to show a little video that I think captures it really quite nicely. This is from a collaborator of mine, Roland Strauss in Mainz. So this is a time-lapse movie of a normal fly. It's quite dark because we've slowed the video right down, but it is actually, the fly can see in this. And the fly's walking along a surface. It's a little bit like the chicken crossing the road, but there's a very large gap there, which for whatever reason it wants to get to the other side. Maybe there's another fly on the other side, there's something going on. It doesn't really matter why it's trying to do it, but it wants to make up its mind, can it do it? So it can judge very, very accurately the size of the gap relative to its body size and whether it can actually manage it or not. Now you can see this is an incredibly accurate, well-considered judgment that it could actually do it. So it's right at the limits of the ability, the physical ability of the animal to actually get there, yet it makes the right decision. Remove one of these stimuli, for instance, change the way that it interprets the distance, it will potentially plummet down and make the wrong decision, or just remove its visual stimuli so it can't see properly, and it'll do something completely different. It basically won't even try. It'll get to there, know there's a gap there, and think, nah, know what? I'm not going to do this. So we've got an animal with 100,000 neurons that can make that decision in real time, incredibly accurately. We'd really like to be able to do that in a computer. Okay. Roland's got a whole series of these videos. I can get in touch with anybody who wants to see them. But I think this is just, it gives a really good example of exactly how well these things can actually think for themselves. And I need to realize that's a loaded term. So we've got this tractable system with conserved function, with excellent genetics. It's autonomous. There's lots of good reasons for wanting to do it. So should we do it? Should we build this virtual fly? Well, I think the idea is that we're going to, and why would we do it, what we're going to do with it. So as I've kind of suggested already, we're going to test our kind of biological understanding. if we can make a simulated fly do what a real fly does, it does give us some confidence that our understanding how the brain is connected is right. If it does something completely different, we've got something wrong. So it's an important test. And that goes for any of these simulations, whether it's a fly brain, a mouse brain, human brain, doesn't matter. Obviously, as I've indicated in that last slide, it's giving us a test of computational complexity. How much can you do with 100,000 neurons? We don't actually know how much a fly can learn. We know it can learn, and we'll work with Barbara Webb in this building. We've actually tried to measure that, and we've measured some of those limits, but we're far from a complete understanding of knowing what a fly can learn when it's given the right conditions and the right incentive to do so. And from that, how you can then potentially use that to change the way you engineer systems. We obviously want to learn how to build bigger brains. We're going to go after the mouse brain, we're going to go after the human brain. But building a smaller one first seems like a kind of good idea. Let's start with something a bit simpler. But obviously we can already start to do things like how do disruptions span these biological levels. So if we can go in and change individual genes, proteins, molecules, or neurons and see how that propagates through a much more complicated system. That gives us an opportunity to do that. And predict the effects of new modifications. That's if you change the system by adding a drug or something like that, what will it do? Will it fix it? So, can we build it? I think basically we're going to. How do we go about it? So I'm going to talk about some of the practical steps now that we've actually taken to actually start to build these things. The first one is with what's called the Virtual Fly Brain Project. You can see where this is leading. Is that we need to actually capture everything that we know about the fly brain. I showed you right at the start of the systems neurobiology, we create these databases that tell us everything we know about something. We want to start to build a virtual brain, we have to do the same thing. Create databases that tell us everything we already know about the system that we can use to connect it together. So the first thing we start off with is the anatomy. So what are the neurons, what are they called, how they connect to each other, where are they, how many of them are there, how often do they change? And obviously we need that to be fit for purpose. So a project that we're involved with started off with what's called the Brain Name Working Group. So it sounds fun. It's an interesting group. And it came across numbers of problems. First of all is if you look over everything that's ever been done on the fly brain, and actually every brain is the same. So people working in other species have exactly the same problems and having to deal with exactly the same issues. So you have a brain region given two different names by two different groups of people working on it. So that's like Edinburgh having multiple names depending on who you speak to. So it's the same place, but depending who you speak to, it has a different name. So how do you tell somebody where you're going? That's kind of easy. You can attach different names onto one thing and you can resolve that and you can deal with it. A much bigger problem is this one, where the same name has been given to two different things. That's like Edinburgh being on the East Coast and Edinburgh being on the West Coast as well. So if you're trying to tell somebody to get to Edinburgh, you could end up in one of two places. And there's problems with that across all sorts of anatomical regions. Boundaries of some regions have not been defined. That's like trying to tell somebody, go to Dundee, but you don't quite know where it is. Or various regions have actually got no names, no boundaries, no anything. Like there is a place called Inverness, but we have no idea where or what it is. Okay? So you've got a bunch of these problems. So what we did was we put this group together to basically just resolve, rightly or wrongly, every single one of these issues as they arise. So try and come up with a standard, what they call a nomenclature, just a naming system, and to make it as compatible as possible. So the group that's put together, I should definitely acknowledge Kazunori and Arnhem, who did a lot of the actual imaging work in this, and then the rest of the Brain Name Committee, really. Started meeting virtually and in various different places to start and put names to these things. And this is the sort of data they started with. These are sections, so slices through fly brains. You think how small the fly is, this is it zoomed up. And it's labeled in different colors so that we can look at the boundaries between different regions and we can go through them and set different things. So actually what we really did was just draw around them. We started as much as possible by saying what are the ones we really know and identify those first. How much evidence can we bring in from other species? Can we start to suggest the name that's been used most commonly in the literature? And can we resolve the problems where a single name has been used to two different parts, and can we resolve that? And that's essentially all been done. So there's now a list of all the brain parts. The first lot, the names have been resolved. There might be multiple names for these now, but we've got a preferred one and a list of all the other ones that have been used in this group. We've got a set where there was a name for it, but nobody quite knew where it was, which was a little bit embarrassing at times. And then a bunch of new regions up here which never had any name, never had any boundary, and nobody knows anything about. So that's actually, this has been an important exercise because this now tells us there's a whole part of this small, tiny little fly brain that we actually don't know anything about, and we're going to have to go back and try and figure out what it is, how it connects to everything else, and what it's for. That's all very well. So that gives us names. That gives us an atlas. We now have an atlas to get around. It doesn't let us build a simulation. It doesn't tell us what connects to what. It doesn't tell us what information goes from part A to part B. It doesn't tell us what genes are expressed there so we can look at the genetics. And so we've then extended that work here with a collaboration with Michael Ashburn and Kahiro Kane's group in Cambridge, primarily with David Sutherland, into a more formal data structure. So we've started to link other information into this anatomy, like, for instance, where this neuron comes from in development. When is it born? That information is now there. What genes or what proteins are expressed in that neuron, that can be linked in. What happens when that neuron switches off? You just kill that neuron off or delete it. What happens to the animal? You can put function in, and so on. And so we've gone from an anatomical atlas that just gives you a picture of where things are to a whole series of relationships about what each of these parts are and how and what the functional importance of these are. So I'm going to skip this on. So again, what connects to what? What's involved in each of the synapses of these things? Where does it come from? And so on. So we can now map all these different things onto it. Looks like one of these. So from a single picture of a single neuron going through a fly brain, you've now got all its history, what type it is, how it connects to everything else, and what it does. So that's what you can actually use to start saying what would happen if you connect two of these things together. So we've now got the first part of a map. And you can go and explore this. This is publicly available. So this is Virtual Fly Brain, Nestor Miliev's work in collaboration with Richard Baldock's group at the Western General. So this now allows you to go through a 3D virtual fly brain. You can see it on the web. This is any web browser pretty much will work with this, some better than others. You can find out where things are, so you can click on the name of a structure which previously would be hard to define. It'll colour it up, it'll find out where it is, it'll give you a text description of it. You can find out where it is relative to some other part of the brain, and you can zoom in and out and colour label and explore other things and see where they are. See how far this goes. I think I'll skip one. So yeah, this is it now going into a different part of the brain, and you can start to see where structures go in 3D and explore them. Why is this not moving on? You can then build the sorts of queries that I was getting onto. If we want to build a real 3D brain, we need to be able to say, right, we've got something going on in here. How does it connect to the rest of the brain? Can you get the circuit diagram for this? And this now allows us to do it. So we can now actually start to highlight what neurons are in here, find out where some other structure that they might connect to, and add it onto a query. So we've got one structure of the brain there, another structure on there, and then go and find how many neurons could potentially be the connection between these. Now, this is an early version of it. We've found 5 neurons that would actually potentially be the connection or a functional connection between these two, and you can get back to the original data that tells you how many there are, where did they come from, what the pictures are, what papers they were published in, and so on. This has grown considerably since this particular example. And it's grown through the addition of this data here. This is Anshen Chang's group in Taiwan have done an extensive piece of work where they've individually labeled those 18,000 individual neurons in the fly brain. So that's a fifth of the entire fly brain. Now, given the fact that not every neuron is unique, 18,000 covers most of the fly brain neuron types that are there. So they've got pictures in 3D of all of them, or most of them. And so they very kindly shared all their data with us. It's sitting in hard drives upstairs. And in collaboration with Greg Jeffries at the MRC in Cambridge, he has developed a system for comparing each individual pair or potential pair of neurons based on shape. So we can then cluster them together, and actually in any part of the brain we can find what all similar neurons are in there. And can you start to look at what's connected to what. So those are in there now. Instead of actually having 18,000 neurons to deal with, you can now look for clusters of ones which are similar, and you can get a measure for how many different types there are of each different neuron, which we can then use to build potential models. That's all in the database. I can skip this quickly, but we can, again, go into the same interface, select a part of the brain, and very quickly do a query for what kind of neurons are in there, and get back any of those 18,000 for any one particular part. And this is a tool for biologists primarily. This is a tool for biologists to go in and say, right, I found something new in this thing here. What's already known? So we're now indexing pretty much every bulk data set that's in the community. There's a couple of exceptions, but most of them are in there. And so you can very rapidly go in and find the pictures and the information about these 3D data sets. Going back with all the original data linked back to the groups who generated them in the first place is all there. So this is then going back out to their database, which gives you all the detail. But it's indexed in one place. So this is as far as we've got in terms of the anatomy. So we've got 100,000 neurons. We've got pictures and information about a lot of them, and a lot of them are being completed with various groups around the world. As I said before, each of these neurons has 50 synapses. Each synapse is a compound processing unit. It's got all these molecules in it that are affected, that are changed by things in the environment. And we reckon it's about 3,000 proteins are in each of these synapses. And if you want to try and represent each one of these in the model, even very, very simply, you're looking at about 15 billion little things that you've got to simulate to get a fly brain. So is it possible? Well, actually, it's been possible for a wee while. You could actually just about do that with the Edinburgh Blue Gene system that was installed in 2005. But me grabbing the Blue Gene for the next 3 or 4 years to build a virtual fly brain, it wasn't likely to happen anytime soon. But we're actually probably going to be able to get access to this new system here. This is the ARM, the chip manufacturer who build a lot of chips for mobile phones. So most of you are walking around with a large number of ARM processors in your pockets. They've, in collaboration with Steve Ferber at Manchester, have developed this new Spinnaker chip, which is actually optimized for doing neural simulations. And so one of these Spinnaker chips can do approximately 16,000 neurons simulations in real time. So one rack, so one pizza box sized piece of kit is approximately one fly brain, depending on the complexity. Certainly one full rack would do the complexity we're after. So there is actually now hardware that's going to allow us to actually build this properly. And we're already starting work with them to do that. So that's as far as it is at the moment. We're starting to build this thing for real, based on the biological data that we've put into these public databases. But I want to finish up on a more practical example. So given our understanding of what we've gathered in the fly brain and how it's connected together and what proteins are expressed there, what proteins are involved in disease, can we actually do something useful with it right now? Let's not wait for 3 or 4 years while we build virtual models and simulations. Can we do something immediately? And there's a number of things we could do, but I've just picked one example for today. And that's kind of triggered by a blog article that came from the Michael J. Fox Foundation website just a few days ago. It's 18th of January. So they actually put something out saying that they're going to start a clinical trial. That's for those of you who know, clinical trials are not cheap. This is a fairly large-scale, expensive study. On the idea of using nicotine patches to stop Parkinson's disease. Why am I interested in that? There's a number of reasons I'm interested in Parkinson's disease, one of which is it's one of those diseases right at the start that is catastrophic on people's lives. So this is the work that we've been doing with Brainwave Discovery. This is a spin-out company based in physiology along the road, where we actually use this information. So we use our understanding of the fly brain to develop new tools that we can use for drug discovery and drug research. So the principle behind it is that we can go from a normal fly on day one and change its nervous system so it's much more like the human. Now, why would we do that? I said early on that a lot of human disease genes are in flies, so why would we need to humanize it? One of the things that we have done is we've looked at how flies respond to human drugs. And 2/3 of them don't work in flies. 1/3 do work, but they don't work as well. And that's just to do with subtle changes. So the genes for the disease might be there in fly, but they're subtly different. They might have similar functions, but they're not identical. And so rather than actually work with the fly genes, what we do is we actually introduce the human ones. These aren't humanized flies. They don't look any different. Well, sometimes they behave different. Basically what happens is we take a small part of the nervous system that we understand, we know what it does in terms of its behavior, and we put a human gene, one or a small number of human genes into the pathways just in those neurons. So you've got a fly which is otherwise behaving completely normally, and a small part of its nervous system now can react in the way that a human nervous system would react to. So we've done a bunch of these. In fact, we've done about 100. And just to show you this actually works, this is an example. So this is a gene called PDE4. There's some collaborators who worked on some of the early versions of this in the audience somewhere. Where in flies, if we look at brain activity, so this little graph here shows brain activity. Flies doing things, we're measuring its activity. Don't need to worry why. In the red line there, we add a human antidepressant called Rolipram, which acts on PDE4. And you see basically nothing happens. And then you get a kind of sluggish, well, okay, I'm going to do something in response to this drug over a period of time. Okay, so it's not exactly, massively exciting, the response you get from the drug. If we then replace the fly PDE4 with a human one, we get a slightly different story. Brain activity again, slightly different scale, drug gets added, and bang, this thing is completely reacting the way we would expect it to do. So this actually inhibits the action of the neuron. So the principle works, okay? What about, we can do other things. So obviously that's just from brain activity. The brain activity measurements are quite expensive, quite slow to do. We also have a number of things where we actually look at behavior. That tends to be the easier way to do it. Although I'm not going to talk about it today, that's where colleagues at Actual Analytics have come in actually developing a lot of the tracking software that we use for measuring these behaviors. So in terms of aging and degeneration, Parkinson's is a degenerative disease. Flies are interesting in that they're mature in 48 hours, they're old in 20 days. So neurodegeneration in flies is quick. If you want to do it in mammal models, first of all, they don't tend to get neurodegeneration very, very easily. They're very resistant to it. Wish we knew why. But you're looking at a very old animal. These are very long, expensive experiments. In flies, 20 to 30 days is what we're looking at. I'm not going to do that. The assay that we actually tend to use is this one. This is a climbing assay. I meant to put this on a loop, so I'll see if I can show it a few times. So these flies have just been banged down to the bottom of a little glass tube about this size, and then you watch them climb back up. So these are flies at 21 days old, 17, 10, 4, and a control. These are the ones that have the human alpha-synuclein variant form. So this is one of the causative molecular reasons for Parkinson's disease. So these are expressing that in a part of the brain. And you can see that by the time you get to 21 days, these are actually much slower than the ones in either the middle or certainly at the far end. And these ones are the same age. So we can actually measure those differences. We do that either by hand. In fact, most of the data I'm going to show you was tracked by Lysimachos by hand, or you can track it automatically as well. So some of the stuff from Actual. So what have we done with these? We've generated a bunch of these. I said we've done about 100 odd human genes. These are just a few of these. So these all start up about here. So just to make the graph look a bit more interpretable, cut off the first part. This is the kind of slow, normal decay you would get with age in a what's a wild-type fly or a normal fly we haven't done anything with. These are the flies that are expressing the abnormal Parkinson's gene, so they're decaying faster. These two strains are both involved in Alzheimer's disease. These are Alzheimer's models, are decaying even quicker, and Huntington's is absolutely catastrophic. In fact, it's difficult keeping them alive long enough. And these are all human disease variants. These are ones that have come from human populations and patients with disease, being put into fly neurons. So can we do anything about it? Most of the drugs that we've tried, I can't show you because they've been done in collaboration with pharmaceutical companies, but I certainly can show you nicotine because it's one of the controls that we use. So given that Michael J. Fox Foundation are interested in nicotine, what do we see with nicotine with this? In fact, there's two drugs on here. First one is, again, there's our wild type, there's our normal decay of the adult animals that gets older. And this is the mutant, or the Parkinson's fly, so it's much quicker. You see eventually they all kind of, they just get old eventually. There is actually there's a problem that accumulates up. But with the nicotine, we do actually see a temporary improvement. It's not as good as a fly which doesn't have Parkinson's. It's not as bad as one that has one either. And this is significant. We see this over and over again. Usually in the region of about 7 days to about 20 days of age. So I've simplified this graph. We normally look at multiple time points along here, but just to show you the effect, this is a bit simpler. I put two drugs on here, nicotine and pergolide, which was one of the old Parkinson's disease. It's been removed from the market because of side effects, but it's still being used occasionally in some other countries. So, suggest this might be kind of interesting. Problem is, it does still collapse. So pergolide's toxic, so it brings the animals down. Nicotine does actually collapse towards the Parkinson's data pretty quickly as well. So this effect is maximal in this kind of trace. Okay, I've chosen these data to make it look as exciting as it can. It rapidly goes back down to the Parkinson's thing. It's not a single cure. This data is a little bit more complex, and this is really what matters. This is looking at the viability indirectly of the neurons that are affected by Parkinson's. And this kind of explains why this effect is potentially temporary. Up here we've got the control brains. So these are the 1-day-old flies. So they have Parkinson's, but it's not affecting them yet. And then at 30 days, they look here. Now, this is kind of difficult to interpret. Actually, the only thing you need to look at is the intensity of the green stuff here and the intensity of the green stuff there, and there's a difference. The difference is essentially the loss of the dopaminergic neurons, the neurons that are affected by Parkinson's. If we treat with nicotine and it was saving the nervous system, we'd expect this to be as green as that. And it's not. It's more like this. If we treat with another drug that does have a previously demonstrated effect on Parkinson's, it would be greener, and it kind of looks as though it is. Now, rather than actually just get you to screw your eyes up and look at this and think about it, obviously we can actually measure this computationally over a large number of samples and actually measure what the amount of that signal that's actually present. So that's the 1-day-old. The bright green brain at the top is this one here. The Parkinson's brain after 30 days has lost most of the tyrosine hydroxylase activity, the green signal. The nicotine treatment hasn't really made an awful lot of difference. It's marginally high, but it's not significant. It could just be random chance. Whereas the normally aged brain is much stronger, and the pergolide treated one is. So in conclusion, I think what we would say is that nicotine may be a temporary treatment. It might help some of the symptoms, but the evidence suggests when you look at an integrated model like this that it's not actually going to treat the root cause of the disease. It is eventually going to catch up with the treatment. And I think that's been seen in several other models as well. There's been several attempts to try and characterize what nicotine does. It doesn't mean to say that it shouldn't be potentially prescribed or the nicotine route or drugs that are like nicotine aren't effective at some point, but we need to be cautious. So in conclusion, I just tried to tell you that full-scale brain models will become reality. That fly is feasible now, so let's do it. Mouse is feasible shortly. Human brain project will start this year and is due to deliver something in 10 years' time, approximately, at least at the scale of a human brain simulation. I also want to describe and share with you some of the work that we've been doing in terms of actually preparing for the virtual fly brain, in terms of developing the tools and the resources towards the model, but also that are useful for biologists. And then finally just end on an example of actually being able to use that to actually get out some insight into drug discovery and drug development. And so with that, I would definitely need to thank all the people who've funded all this research: BBSRC, EPSRC, MRC, Wellcome Trust, European Union, and more recently the National Council for the 3 Rs, and all my collaborators, most of which are mentioned on here. And finally, thank you all for coming out this evening and for your attention.
So we've got time for a couple of short questions. Do indicate who you are.
Thank you. So you mentioned that the goal of the Human Brain Project was to simulate the human brain. There are two questions, really. One is, how close do we have to simulate the components in order to do this? I mean, do we have to build really accurate neurons, or could we build them out of something simpler? And secondly, when would you tell me that I succeeded?
For the human brain, not the fly brain. Ah, right, okay. Flies would be better. For the human brain, do you need to build real neurons to come up with a simulation that could behave convincingly as a human, presumably over the internet or something like that? Obviously, no, it wouldn't need to be real neurons. If you want to simulate the effect of a disease and how that propagates all the way up, then I think you would need to have something that is approximately that level of complexity. And that's an ongoing argument in the area, is do you need to go down to that level of complexity? And I think it depends what you want to do. If you want to bring something that can behave and interact and be reasonably intelligent, then potentially not. But if you want to simulate its disorders, then yeah, you do. It's probably more the latter than I'm interested in. So I would focus on the molecular level.
So is there any single neuron in the fly brain that if you destroy it, that is essential to its function, that if you destroyed it would degrade significantly the function of the fly?
One neuron, no. Two, potentially, that you can measure. And there's more than one pair of neurons that you can switch off and do. There's there's circuits. The giant fiber neurons are probably the best known ones. And those are involved in, if you go and try and swat a fly, they're involved in a very fast reflex from the visual system to the middle, I think it's middle legs, that will launch the animal in the air. So before it's actually really seen your hand coming towards it, it's already airborne. That's why it's quite hard to hit flies on the surface. So if you switch those neurons off, yes, you're going to get a measurable effect. You can hit the fly faster. But there's also a whole series of other ones. There are ones which actually regulate learning and memory processes that help propagate signals around the neuron, around the brain. So those are examples of ones that are very, very predefined, but there are equally other ones which are much more variable in terms of the number of the neurons and what they do and how they connect up.
Henry Thompson from here. So in your answer to Peter's question, you got close to something that intrigues me about this talk, which is the title was about finding answers in the mind of a fly, and you spent your talk quite carefully talking about neurons. But in answering Peter's question, you sort of came close to having to take a stand on the relationship between the two, which, you know, do flies have minds? Do you need to get to something layered on top of the brain before you can start talking about mental disease?
We don't know how much flies have got. The instant answer would be no, of course not. They're far too simple. But actually, once we start teasing into the fly, it seems to be capable of doing more and more things. There are limits. There are things it can't do, but we don't know the full set of those limits or what motivates it yet. So it's yeah. I mean, the title was kind of, you know, I used those words deliberately. It wasn't the first choice of title, but I was overruled on that one. It had 4 of the same words in it. But yeah, It's an unknown one. We don't know the answer. I don't think we'll know anytime soon either.
How distinct would your full-scale model be from life in a pure sense? So if you actually succeeded in making a full-scale model of a brain, how similar would it be to a living being's brain?
Well, that's the test, isn't it? That's how you tell whether you've done something useful or something realistic. And a lot of that's going to come down to how you connect it up. You know, how do you actually get inputs into it? And that's not an easy thing to do. There's a lot of large-scale simulation projects have struggled with exactly how do you get sensory information into a simulation and how do you get motor information out. For the fly, as is said right at the start, those connections are better understood. And there are at least reflex simulations now that can show you how a fly can self-right itself, yet without that implicit instructions being in the simulations. So there are groups who have actually started to see those kind of emergent properties come in out of these simulations. And I think that's really what you would test. Can it do something you didn't actually build it to do, but the real animal can do as well?
So thank you very much. Thank you, Principal. Well, how good was that? Well, it's fab. So thank you very much indeed. I always find it fascinating to come to these inaugural lectures because I learn such a lot. Now, I'm a chemist, so I got all excited at the beginning there when you talked about the chemicals. But in fact, it was much, it was just so interdisciplinary. You just encompass so many different interesting aspects of the work we do here at Edinburgh and, in fact, with all your collaborators. And then in the questions, we then went into what's the difference between the mind and the brain. So we're getting very heavy there. So thank you very much. Before I ask you all to thank again Douglas for that wonderful lecture. We heard about the influence of nicotine, but now I think we've got a chance to study the influence of alcohol, have we? So please join us all afterwards, and you can speak to Douglas afterwards out here for a drink. But first of all, thank you. Thank you, Douglas, and please join me in thanking him very much. That was fabulous. Thank you very much.
This production is brought to you by the University of Edinburgh.